Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. I hope you had a great week. Before we get started, remember that Let's Talk Micro is available on Apple Podcasts, Amazon Music, Spotify, Google Podcasts, Stitcher, TuneIn Radio, Overcast, Pandora. Whenever you listen to your podcast, you can listen to Let's Talk Micro. I am also on Instagram as Let's Talk Micro, no apostrophe, and on Twitter as Let's Talk Micro 1. So go ahead and follow. I like to post pictures of organisms, some nice pictures that I take. So last week we didn't have an episode. There was no episode. My apologies. I had my birthday, school, work, many things going on, so I couldn't record an episode. And then on Instagram, one of the followers suggested that because I posted a picture of Pastorella multocida, which happens to be my favorite organism. So the follower suggested that I make an episode about it. So I'm going to take that into consideration, and I will be making an episode about it. Once I make it, I will let you know either via Twitter or Instagram when the episode will be up. But it's a pretty fun idea to talk about my favorite organism and why is it my favorite. And like always, if you have any suggestions, go ahead and just hit them. Hit them via Instagram, via Twitter. I also have an email address, which is letstalkmicro, no apostrophe, @outlook.com. If you are on a medical technology program, like a medical lab sciences program, an MLT program, if you're on a microbiology association and you want me to come on board and talk about my podcast, talk a little bit about the profession of the medical laboratory sciences, go ahead and either email me at letstalkmicro@outlook.com or hit me via Twitter or Instagram and let me know. I have a very busy schedule, but I will definitely try to work something out. This is the goal of this podcast. I want to promote microbiology. I want to talk micro. And I want to talk to people, bring awareness to this profession of the medical laboratory sciences and such a great work that is done. So on the last episode, I introduced the anaerobes, right? I started talking about them. I went over classifications of the organisms based on their tolerance to oxygen. Right, I discussed that there are aerotolerant, microaerophilic, facultative anaerobes, and obligate anaerobes. And if you haven't listened to this episode, please go ahead and do so. This way you can have a refresher. I want to make clear that when I'm talking about anaerobes, I am actually talking about organisms that do not grow in the presence of oxygen. So this group of organisms that I'm talking about, they do not grow in the presence of oxygen. And why is that? Well, I talked about it on the previous episode, but they lack the superoxide dismutase and catalase, which are enzymes that are required to break down reactive oxygen produced during respiration or aerobic metabolism. So let me say them again. They lack superoxide dismutase and catalase, which are enzymes required to break down reactive oxygen produced during respiration or aerobic metabolism. Oxygen has a high affinity for organic compounds containing nitrogen, hydrogen, carbon, and sulfur, which interfere with normal biological activity. So these organisms, they can't protect themselves against this. So they need an absence of oxygen to grow. So in order to recover anaerobes, we want to make sure that we actually do the proper collection, transport, and source. These organisms from the get-go, like I said, they do not grow in the presence of oxygen because of lacking, the lack of these enzymes. So we want to make sure that we actually have the proper collection, transport, and source. Not all sources are appropriate for anaerobes. So let me read this from the American Society for Microbiology. They say that the collection of specimens and swabs should be discouraged for anaerobic recovery. This is because swabs have a small surface area and are prone to pick normal flora. providing an inaccurate result of what the pathogen should be. Specimens should be transported at room temperature, and then proper sources include deep aspirates and wounds, or specimens that are surgically collected. Unacceptable sources are throat swabs, urine, and superficial areas. So as we think about this, we're like, why, let's say, a superficial area is unacceptable? Well, think about it. These organisms, they cannot grow in the presence of oxygen. So in surface areas, they are exposed to oxygen. So it makes sense that they will grow in deeper areas. So good sources are like when you get like a body fluid, a good source that is collected surgically from an interior source. So that will be a good source. But going back to what the American Society for Microbiology said, sometimes you get in the lab, you get a swab that is both for anaerobic and anaerobic culture. So by the time you're done plating and everything, you expose that swab. So it might affect the recovery of the anaerobes. So you need to make sure that you have the proper source. So now that we have picked the proper source, what about the incubation requirements? Right? Like I keep saying, they cannot grow in the presence of oxygen. So how do we achieve this? Well, you have several methods that are made by manufacturers. What they use, what it's used depends on the size of the lab. Anaerobic bags and jars are widely used by the labs regardless of the size. And then for smaller labs, the bags and jars are more cost effective than having a big system like the anaxomat. Especially if you have a low volume, it makes sense to have the bags and the jars. And I'm going to be talking about these systems. They typically have a sachet that absorbs oxygen and produces CO2. So definitely if you find yourself working in a smaller facility, it's going to be bags and jars. They are relatively inexpensive and they don't take a lot of space depending on their volume. So you can justify having those bags. When you get to a large system like the anaxomat, you need to maybe justify the cost. So you might see that in a large facility with plenty of volume. Like the facility that I work at, they do have a pretty huge volume. So they use the anaxomat. And of course, you also have bags and jars to complement it. Because depending on the volume, if you're working on the bench, it's easier to use the bags. But I'm going to be talking about these systems and how they work. So let me start with the BD Gas Pack Easy system. Now, this system, they have no relation to the podcast. So it's just me talking about them from what I have experienced. So this system is with the bags and sachets and so forth. So it has a sachet that contains inorganic carbonate, activated carbon, ascorbic acid, and water. Upon exposure to the air, the sachet is activated and it reduces the oxygen concentration and produces CO2. They come in smaller sizes where you can place a pouch, with 1 to 4 plates. Then there is a larger size that you can use for a jar, and you can fit about 12 plates. It comes with an indicator that changes color if the bag or jar was not properly closed. For this system, this changing color is from white to blue. Let's say that you are doing an air tolerance test, right? That we talked about when I introduced the anaerobes. You have an organism that's growing on your CDC, on your shalers. Based on what you know about micro, you know that they are facultative anaerobes. So, your staph, you can have streps, enterobacteriales growing on anaerobic media. So you want to make sure that that organism is a true anaerobe. So you do your air tolerance test. You go ahead and sub your colony to 2 blood plates. Sometimes in some facilities, and it is recommended that for the aerobic portion you sub a chocolate or add a chocolate. So make sure that there's not a Haemophilus that you're missing. As you know, Haemophilus does not grow on blood agar. It can, but under certain conditions that once we get to that point, I'll talk about it. So you do that, and then this bag system, it's pretty convenient for that. Typically on your bench in the drawer, you have your bags, you have your pouches, your sachets, your indicators. So you just grab the bag. Once you subculture your plate, put it in the bag, put the indicator, and put the sachet. Make sure you close it properly, right? Typically, once you close the bag, some people with their marker, they press on it, and then you incubate it for 2 days, sometimes a little bit longer. So I talked about this indicator, right? So this says that changes color if the bag or jar was not properly closed. So, and this system is from white to blue. So that means that if 2 days later you come in and grab your bag and you see that the indicator is like a little pill enclosed in plastic. If that little pill, it's like a little tablet, it is blue, that means that you did not close your bag properly. And unfortunately, it happens. Sometimes you're going too fast with the jars. You don't realize you're like digging for things in the incubator. Accidentally, you open that jar, you don't realize it. And then when you go in, you see the blue color pill in this case. And definitely that's not good. So it's definitely a very easy, very user-friendly method for anaerobes. And like I mentioned, you want to incubate it for 1 to 2 days. And once I get through these systems, you're going to see that sometimes, maybe having it for 2 days is better, because the time that it takes to achieve an anaerobic environment, it varies with the systems. So sometimes, you might not have enough growth. And then if you're on the bench, you encounter this organism that you're doing the air tolerance on maybe at 1 o'clock, let's say you're on day shift. And then you put the bag, and then whoever is reading anaerobes the next day, they come in at 6, they take out all the bags, they're sitting there. So you might not have enough growth. So you have to keep that in mind. So depending on what system you use, you might have better growth at the day mark. If not, you need to wait the 2 days to make sure that you have enough growth, enough isolation to perform the ID, which for that you're going to need different volumes of the organism, different amounts. So now that I talked about the BD gas pack easy, as an EZ system, let's go ahead and talk about another system. You have the Mitsubishi Gas Chemical Anaerobe Pouch Anaerobe and Anaeropack Anaerobe. This system also has a sachet that absorbs oxygen and produces CO2. It has an indicator that changes to purple if the bag or jar was not properly closed. In the bag or pouch, you can place up to 3 plates. And in the jar with the large sachet, you can place 12 plates. So once again, you can place up to 3. It's always best if you have 2 of them. And then typically with this system, you put, let's say that you're only trying to isolate one organism. You're doing the air tolerance challenge on one organism. Typically you get a plate, like a blank plate to balance it. And then you incubate both in the bag. So in the bag or pouch, you can place up to 3 plates. And in the jar with the large sachet, you can place 12 plates. So once again, with this indicator, it's going to be purple. It's pink. And just like the other one, just like the BD Gas Pack Easy system, it's like a little tablet enclosed in plastic. So it's just as user-friendly as the BD Gas Pack system. You do your air tolerance, get your organism, put it on the plate, put it in the little bag. In this case, if it's only 1 plate, put a second plate, put the little sachet, your indicator, make sure you close it, and a 2-day open date. And that's it. And then the third system that I'm going to be talking about is the anoxomat system. So this one, like I mentioned before, it's more cost effective to have it in a large facility. It takes more space than the other ones because if your volume is not great and like in a smaller lab, you can find an area where you can store your little jars, your pouches, your bags. This one involves an actual tank. So you need more space and it's more expensive because you have to replace the tanks. So for this one, it's best if you work in a large facility. However, once I talk about the systems, you're gonna see that this is my opinion. From what I see and from what I'm gonna tell you, I think you might draw the same conclusion that this is the best system out of the three, out of the previous two that I talked about. So with the Anaxomet system, it has a computer which guides the process. You attach the jar to a tank that has the anaerobic mixture, which is 80 to 90% nitrogen, 5% hydrogen, and 5 to 10% CO2. It does 3 cycles where the contents in the jar are evacuated and replaced with the gas mixture. And there is a palladium catalyst that removes any remaining oxygen after the 3rd cycle. So this system is a little more involved, it's a little more time consuming than the other two that I talked about. Like in the other two systems, you sub, put in the plate, put in the bag, pouch, indicator, close it, incubator, and you're done. Whereas with this one, like I said, you attach the jar to a tank that has that mixture. So there's a series of events going on here. And the Anaxomet is going to perform a series of checks that if it fails, then it's not gonna end the process with the jar properly sealed and that anaerobic environment achieved. There's many places. For example, if you place the lid on the jar and there's not a good seal, sometimes on the lid there's like a rubber band, in a sense like a rubber band. If that is maybe loose, if it doesn't seal properly, it's gonna detect like a leak, so it's gonna stop the process. It's gonna give you an X. As it goes through the series of checks and it passes, it gives you like a green check mark. So if something fails, it's gonna give you a red X. Also, if the jar is not connected properly, it's also gonna fail it, giving you that red X. So you have to make sure that, and it gives you the proper way, it gives you a diagram of how to properly attach the lid. Make sure that you remove any excess air that's in there. And if you fail to put like the catalyst pouch, that would also give you a fail X, a red X. So there are more things that you have to pay more attention. I mean, like anything, you always need to pay attention to your work, but with this one, you have to make sure that you follow this series of steps. So your jar can produce that, not only pass with the green check mark, but produce that anaerobic environment for the organisms, which is what you're going for. And with the catalyst, the palladium catalyst, when I started using the system, this catalyst, they were reusable. Like you actually had to, once you were done with them, when you opened your jars, you have to put them in an incubator and raise the temperature. I think like about 100 degrees or so. But nowadays, we're moving more towards like there are disposable catalysts, so that's good. So this was one of the cons of the system, that you had to heat this catalyst, like you had to reactivate them. But now that we have the disposable ones, it's much better. So having said all this, I'm gonna mention some things that I already said and add some new ones. So these three systems, I'm gonna compare them. Having said that, once again, they have no relationship with this podcast at all. This is from what I've seen, from what I read. They have nothing to do with this podcast. So let's start with the easy. How long does it take to achieve that anaerobic environment, right? So basically, you want to make sure that the oxygen is removed because it's harmful to these organisms. So with the easy system, it takes about 2.5 hours to achieve an anaerobic environment. The Mitsubishi, it takes 2 hours. So these organisms, they're already sensitive to oxygen, so it takes 2 hours. So that means that when you're using these systems, you have to make sure that as soon as you seal that bag, put it in the incubator. You sub that plate, put it in the bag, seal it along with the indicator and the pouch, and put it in the incubator. Sometimes, large labs, when people are busy, they tend to go ahead and they sub out their plates, especially, when you're in the processing area, that you are processing the aerobic and anaerobic cultures. And some people sub their plates, put them in the hood, or put them in some sort of bin or receptacle, and then they might leave them there for a while. Sometimes, busy days, they might go on break and leave the plates out. So you imagine how the recovery of an anaerobe can be affected when you are inoculating a sample on a plate, and then that plate sitting there for an hour. Then you put it in one of these systems, and it takes another 2 hours. So that's a total of 3 hours that organism was exposed, in a sense, to oxygen, or it took 3 hours for that oxygen environment to be produced. So the recovery of the organism might be affected. So you have to keep that in mind. And then when the anoxaman, it takes 10 minutes. That one is definitely better because you can sub out your plates, put them in there, and in 10 minutes, you have an anaerobic environment. And it definitely shows. Let me say this, it definitely shows. I remember that one time I was working a culture and I had an actinomyces. And actinomyces is air tolerant, so it can grow aerobically, but definitely at a slower pace. So it likes to grow anaerobically. And this culture, it had been a few days since it was set up. So I had an actinomyces. However, I'm talking about the pre-Moldy-Tough days, where those of you that know the Moldy-Tough, and I will do an episode about it, with the Moldy-Tough, you don't need much to get an ID. But if you're doing, let's say, like the Vitek, the ANC card for an identification, for actinomyces, you need a 3.0 McFarland standard. So for that, you need a lot of organisms. I knew that was an actinomyces. I was suspecting it, so I wanted to rule it out. I went ahead and subbed my plate, put it in an oxamat jar. The next day, beautiful, strong growth. Those 10 minutes, definitely, it's creating that environment is such a big help for the organisms. And in my opinion, it definitely maximizes the recovery. So definitely those are the cons for the Easy and the Mitsubishi, that it takes a long time. 2.5 hours for Easy, 2 hours for the Mitsubishi to achieve an anaerobic environment. And if you combine that with other factors, like sometimes, maybe techs leaving the plates out, or for longer periods of time, if you go ahead and subculture your organism, maybe late in your shift, then the next day someone comes in at 6 a.m. It hasn't even been 24 hours, and they put the bags out there and they're just sitting there for hours. So your recovery is affected. Now, having said that, the pros are that they are, more cost effective for smaller facilities, and they're easy to store. They might not take much space depending on the volume of your lab. With the anoxamat, a pro is that definitely those 10 minutes, which at the end it works better for the organism. So I think that, in my opinion, it outweighs the cons. Which the cons might be that, yeah, it's a little more time consuming because you have to make sure that you follow some steps, so all the checks pass, and then anaerobic environment can be achieved. And then also you have to replace the tank. So that's another thing that, depending on if you're on an off shift, and then you have to call someone to bring it, and it's heavy, and you might lose some time doing this process. But I think at the end of it, it's worth it because the recovery is better, the organisms grow better, and this is the whole point of why we do this for the patients, to make sure that we put out there the best results so our patients can get the proper treatment and recover. And that, my dear audience, it's the end of this episode. I hope you enjoyed listening more about anaerobes, because I definitely enjoyed talking about them. Like always, continue your motivation, continue bringing that passion. I cannot mention this enough. This is such an amazing field. This is such a great work that we do working with these organisms, making sure that we provide the best IDs, best susceptibility, so the patient can get the proper treatment and recover. The world of the microorganisms is such an amazing one. So please continue staying motivated. And once I do the pastorella episode, I will let you know. In the meantime, stay safe, stay motivated, and of course, continue talking micro. Until the next time, bye.